Well, good morning, everybody. And thank you very much, Richard, Ramon, and Edward, for inviting me to Barcelona, particularly in this wondrous hospital, where it must be an aesthetic pleasure to be admitted. You would like to be sick here. But the challenge they have been given is to defend a hypothesis. So the way I'm going to do it, I thought, is to clearly distinguish as we go along the reasoning facts from hypotheses, so that you can make up your mind and decide what can possibly be true. And the other disclaimer that I have to make immediately is that this is just one of the potential mechanisms, which is neither exclusive nor incompatible with the other mechanisms that have been alluded to by Richard, the diuretic, the blood pressure, the vascular effects, and so on and so forth. So now, this is my disclosure. Metabolism is complex and sometimes complicated. So I thought we could start from the fundamentals of the glucose-insulin feedback mechanism. And what you have here is the beta cell, which under normal circumstances secretes just how much insulin is needed and in appropriate time course, and also suppresses alpha cell activation to produce glucagon. So the combination of these two results in stimulation of uptake of glucose by muscle, adipose tissue, and a few other organs, and inhibition of endogenous glucose production, mostly by the liver, but possibly also by the kidney, as well as suppression of lipolysis with liberation of free fatty acids from the triglycerides that are stored in adipose tissue. And by doing that, you keep both the fasting and the postprandial glucose levels under normal limits. And in normal circumstances, there is very little urinary glucose excretion. Now, the one thing that is important to remember here is that the dose response for stimulation of glucose uptake or suppression of endogenous glucose production or lipolysis are very different. It takes much less insulin to suppress productions than to stimulate uptake of glucose by tissues. And this is not without consequences. Now, diabetes is the condition where there is something wrong with the beta cell, which could be only functional, or it could be a loss, a major loss of beta cells, such as is the case in type 1 diabetes, whereby the inhibition of alpha cell activity is released, and therefore there is an excess of glucagon, and then the stimulatory effect of insulin on glucose uptake is reduced. The inhibitory effect of insulin on endogenous glucose production and lipolysis also are reduced. And because of an increase in glucagon, there is a further augmentation of endogenous glucose production. And I call it endogenous because it's mostly liver, but it could also be kidney. And from the SGLT2 science, we are learning that the kidney is quite important. And I should say that not very much is known, surprisingly, about kidney metabolism. There are plenty of experiments in rats, mice, and lower vertebrates, but very little in man. So the consequence of this is that glucose levels will increase, both fasting and postprandial, and there will be a variable amount of urinary glucose excretion as a result of exceeding the renal threshold. Now, it is important to point out that SGLT2 inhibitors have been tested in patients with type 2 diabetes. There are also data emerging in patients with type 1 diabetes and a little bit, as Richard alluded to, in non-diabetic patients. But in type 2 diabetes, the stage is already set for lipid oxidation at the whole body level, which is what these data illustrate, is increased precisely because there is insulin resistance or insulin deficiency. So, for example, in this study, if we measure whole body lipid oxidation in the fasting state or following a mixed meal, you see that it goes up after a meal because the meal contains lipids. But in both fasting and post-meal conditions, lipid oxidation is increased in comparison with individuals with normal glucose tolerance using the exact same technique. Yes?
Are these both groups matched by BMI?
Yes, they were. So we already have a stage set for reduced glucose utilization because of the insulin resistance, which is a hallmark of type 2 diabetes, but concomitantly, because of the ability of insulin to suppress lipolysis, increased dependence for energy production at the whole body level on the oxidation of fatty substrates. And so this is what now goes under the name of metabolic flexibility, which used to be called substrate competition. Then a better name was invented. And this is the reciprocal relationship in vivo between lipid oxidation and carbohydrate oxidation. So if we take the mean values of those four groups that I just showed on the preceding slide, you see that there is a very good reciprocal association between the two, such that when lipid oxidation is down, carbohydrate oxidation is up, and vice versa. And this reflects the flexibility or the ability of tissues to use both substrates for energy production. Practically only the brain, and perhaps not even the brain, is exclusively dependent on the utilization of glucose, but most other organs can flip-flop between fatty substrates and carbohydrate substrates for the production of energy, because energy must be produced for cell survival. Now, here comes the setting with hormones and substrates. If lipolysis is less suppressed by insulin because of insulin resistance or insulin deficiency, the circulating fatty acids will be increased to a variable extent, but they will be increased by mechanism. This is not a hypothesis. Also, there will be less insulin and more glucagon, precisely because of the mechanism described in type 2 diabetes. And we've known for many, many years that the ratio between insulin to glucagon at the prehepatic level is the main hormonal signal for the liver to use substrates, particularly glucose. So you have more fatty acids fluxing to the liver under conditions where glucagon is in excess and insulin is deficient relative to one another. Now, it should be recalled that the liver normally lives off lipid substrates. It runs on free fatty acids. If you measure the respiratory quotient across the splanchnic area, something which we did ages ago, it's 0.7, so it's close to almost exclusive utilization of fatty acids. So if you provide now an excess of fatty acids in the context of a reduction of this ratio, the liver must dispose with this excess fatty acids in some other way. One way is to increase de novo lipogenesis, which leads to the accumulation of triglycerides within the liver. But the other way, to dispose with the excess acetyl-CoA derived from the beta oxidation of the activated acyl residues, is to channel the acetyl-CoA into ketogenesis. Again, this is basic textbook physiology. And so here you have the conversion of acetyl-CoA into acetoacetate via this enzyme, which is very familiar to all of you because it's the rate-limiting enzyme in the synthesis of cholesterol, right? Except that this is the mitochondrial isoform, which carries out this function, whereas the cytosolic isoform is responsible for the synthesis of cholesterol. And this may explain why, in the phase 3 studies of SGLT2 inhibitors, it's consistently been noted that there is an increase in a slight but statistically significant increase in the circulating levels of LDL cholesterol. This could well be the mechanism. You provide an excess of acetyl-CoA to the cytosolic HMG CoA, and that leads to an increase in squalene and all the precursors of cholesterol. And this was initially a concern in the mind, particularly of the cardiologists, because the cardiologists have a mindset that whenever they see an increase in LDL cholesterol, that's an increase in cardiovascular risk. Okay? So here we go, that ketone bodies, acetoacetate, which is then reduced to beta-hydroxybutyrate through the dehydrogenase here, which is expressed obviously in the liver. And, for example, beta-hydroxybutyrate, which is the dominant metabolite of ketogenesis in the circulation, increases the production. Now, another characteristic of the human liver is that it can make ketones, but it cannot use it because the rate-limiting enzyme, which is called SCOT, succinyltransferase, is not expressed at very high levels in the liver. So the liver is only left with the possibility of exporting the beta-hydroxybutyrate into the circulation. Now, this should also occur in the kidney, but there is very little human studies actually documenting this. But the pathway is the same. So beta-hydroxybutyrate should increase. And does it increase? And the answer is yes. This is again the two groups of people, about 65 patients with run-of-the-mill, well-controlled diabetes here, and 25 non-diabetic individuals here. And you see that in parallel with an increase in lipid oxidation in patients with diabetes, there is an increase in both the fasting and the post-meal levels of beta-hydroxybutyrate. And this is statistically more than what is seen in people with normal glucose tolerance. So far, nothing outside of straight physiology. And if you now also plot the mean levels of plasma beta-hydroxybutyrate concentrations in the metabolic flexibility plot, you have that in parallel with this reciprocal association between lipid and carbohydrate oxidation, you have an increase in beta-hydroxybutyrate when lipid oxidation is increased. Just as simple as that. Now, what happens when you give an SGLT2 inhibitor? And the key element here is that you get glycosuria. These are the data that we published a few years back, where, interestingly, the first pill ever of an SGLT2 inhibitor was given here, half an hour before time minus 180. And we collected the urine very carefully, and we measured the glucose excretion. And you see that in comparison with the baseline, on treatment, and these are chronic data, there is already detectable glycosuria in the fasting state, which is obviously exacerbated following the mixed meal. Now, if you add up all the glucose that was excreted under these conditions in the 8 hours of the study, 3 hours of fasting and 5 hours post-mixed meal, the amount of glucose that was leaked into the urine is large. And if you extrapolate now to 24 hours with 3 meals, you end up with numbers anywhere between 60 and 120 grams a day, which is a glycosuria that you only see in decompensated diabetes, in DKA admitted to the emergency department, and transient, because then hopefully there is treatment. So the key thing here is that the kidney gets a kick in the back. It's not just a small amount of glucose leaking into the urine. It's massive glycosuria. And this occurs every time that you give the pill. So every day you take the pill and you spill out this amount of glucose, you stop the pill, and within 24 hours, this phenomenon disappears. So as Richard was saying, it's absolutely logical to hypothesize that the kidney must be responding to this insult or to this change, which under normal living conditions, it really never sees. Now, the first consequence of that is a reduction in plasma glucose concentrations. That's because you take the glucose pool, which is expanded in diabetes, you open the tap and you have glucose leaking into the urine, and so you are emptying out the pool of its contents, water and glucose. And consequently, you have a reduction in both fasting and post-meal plasma glucose concentrations in response to even the first or any subsequent administration of an SGLT2 inhibitor. And this is, again, class effect, no difference. And again, what is amazing is that we could measure the reduction in plasma glucose, in fasting glucose levels, literally a couple of hours after giving the first pill. Very rapid effect. There is no pharmacokinetics that can explain this. This is a very rapid effect, which makes some people think that there is also an independent mechanism acting on the liver. But be it as it may, what you have is a reduction in plasma glucose levels, and that's why these are anti-diabetic drugs, right? Then what also goes with that is a reduction in plasma insulin concentrations, because under all circumstances, with or without an GLP-1 potentiation, with or without drugs, the most important driver of plasma insulin levels or insulin secretion is the plasma glucose concentration. So if you let plasma glucose levels fall without doing anything directly on the beta cell, you have a reduction in plasma insulin levels, both fasting and post-meal. And this was initially unexpected and still partly unexplained. There is also a concomitant increase in the circulating levels of glucagon. Again, both fasting state and post-meal, when glucagon levels are normally suppressed. So you're back to the ratio between these two, and this was calculated as the molar concentration ratio between insulin and glucagon in the prehepatic blood. There are formulas that can be used to estimate this. And obviously, because insulin was going down and glucagon was going up, you have a reduction in the insulin to glucagon ratio, again, both baseline and following the meal. So you are back to the scheme that we described previously. So you expect that lipolysis would be unrestrained, so you measure plasma free fatty acids, and you see that FFA levels are increased fasting and less suppressed during the meal. All of this is statistically significant, but you can find it in the publication. And this is the balance now that you can build of the carbohydrates. So you have, before treatment, the contribution of oxidation, again by whole body indirect calorimetry, and non-oxidative glucose disposal, which is mostly glycogen synthesis in the pre-treatment state. Now you come in with your SGLT2 inhibitor, you expose the body to loss of carbohydrate via the glycosuria, and correspondingly, there is a reduction in both carbohydrate oxidation and non-oxidative carbohydrate disposal. So now you add on top of this an SGLT2 inhibitor, and what happens is that you are increasing lipid oxidation, both in the fasting state and post-meal, over and above the status of diabetes that I described, simply because you are reducing the insulin to glucagon ratio further with the SGLT2 inhibitor, freeing up lipolysis and increasing circulating free fatty acids. So you can now plot this on the same scheme, very simple scheme, saying that there is nothing directly on the beta cell, and this has been documented experimentally. If you take a beta cell culture or even human islets and you challenge them in vitro with an SGLT2 inhibitor, nothing changes. Then there is this decrease in insulin, increase in glucagon, therefore endogenous glucose production is stimulated, lipolysis is unrestrained, and there is a little bit less carbohydrate utilization because you've lost the carbohydrate. You've created a state of relative carbohydrate deficit, whereby the other substrate, the alternative substrate, free fatty acids, is utilized instead. And now you have massive glycosuria. Therefore, the plasma glucose concentrations are reduced, and the postprandial glucose levels are also reduced because you are just leaking out the glucose through the kidney. And if you now look at lipid oxidation, again, in the patients with type 2 diabetes, but also in the subjects with impaired glucose tolerance or normal glucose tolerance, where we could do this, you see that the same occurs in non-diabetics. There is nothing specific to diabetes in the mechanism, so far, in the mechanism of action of SGLT2 inhibitors. This has been done already in the phase 2 studies when non-diabetic individuals are exposed to the drugs to see the pharmacokinetics and the pharmacodynamics. And this is because the SGLT2 co-transporter is expressed in everybody, regardless of the level of expression or function, but it's in everybody. It's an evolutionary kind of mechanism to conserve glucose, water, and sodium under normal living conditions. The only difference is that because of the lower glucose levels in non-diabetic individuals, glycosuria is less, and therefore the entire sequence that we've described so far is attenuated. And therefore you see that this increase in whole body lipid oxidation tends to be smaller in non-diabetic individuals than in patients with diabetes. Now, if you imagine that this is over a range of severity of hyperglycemia and diabetes, you can modulate these bars such that in decompensated diabetes, these effect would be higher, in well-compensated diabetes it would be smaller, and so on and so forth. So far, everything follows. And if you measure circulating beta-hydroxybutyrate after 4 weeks of treatment of an SGLT2 inhibitor, you see that in comparison with the pre-treatment values, which are suppressed during the meal because of the increase in insulin, that's the normal response, you have relative hyperketonemia after treatment, both at baseline and post-meal, as a reduced suppression of free fatty acids. Now, I should mention now, at this stage, something which even clinically is quite important. What is plotted here is the mean values, and these are standard errors of the mean of beta-hydroxybutyrate levels. But these concentrations I found when I measured them in this study and in other studies are not normally distributed. They are non-normally distributed. So there are a few people in whom even just at baseline, you find beta-hydroxybutyrate in the millimolar range. And when you give an SGLT2 inhibitor, in some individuals, and I will show you a little bit of the data, you see that beta-hydroxybutyrate shoots all the way up into the millimolar range. And initially, we thought this was an analytical mistake. So we went back and reassayed the samples. But it is true, and it's been found in other studies likewise. So it's not necessary to think that you need, on average, a big increase in beta-hydroxybutyrate to produce any hypothetical biological effects. They may be occurring only in those patients in whom, for some reason, and you can already argue that that reason must be insulin, either deficiency or resistance, because insulin is the major suppressor of ketogenesis, as is the case for lipolysis. And I reminded you of the dose response, which is much tighter, shifted to the left for suppression of lipolysis compared to promotion of glucose uptake. So it's not necessary to think that every diabetic would respond in this fashion. This is now the excretion, and this is a point that was touched by Richard, and this is where I raised my hand to disagree, Richard. In resting conditions, ketones, as I will show later, are taken up by multiple tissues except skeletal muscle. Skeletal muscle under resting conditions uses very little ketones. Ketones are only increased in uptake if you exercise. So the reason why you have this accumulation of beta-hydroxybutyrate of ketones in the circulation is not because there is deficient uptake by some organ. Certainly not skeletal muscle, but very probably not even the heart, which was your hypothesis, because the contribution of the heart, 300 grams versus 28 kilos of skeletal muscle, is too small to induce any change in the circulating levels. Not only that, but contrary to what was hypothesized initially, when we measured the excretion of beta-hydroxybutyrate through the urine, we found that this excretion, in parallel with the increase in levels, was increased, not decreased. So again, the accumulation of ketone bodies was not due to reduced excretion by the kidney. This can happen if the patient is dehydrated and therefore the GFR is cancelled, as is the case in DKA. And you know very well that the treatment of DKA involves, first of all, rehydration to restore kidney function, which will then also help eliminate the excess ketones that have been accumulated during the ketogenic phase. So ketones, by and large, accumulate because they are produced more, not reduced utilization. Richard. All right, now, one problem in my mind and one objection that has been raised so far is that, well, what happens if I, at the same time as stimulate this whole sequence of events with an SGLT2 inhibitor, I come in with something that stimulates insulin release? And it was very handy to lay my hands on the samples from the Duration 8 study, where what they did, this is a trial that has been extended to 1 and 2 years now, where they randomized people, about 250 per arm, to either dapagliflozin and placebo, or exenatide once weekly and placebo, or the combination. And as you know, this has been published in The Lancet 2 years ago. This is what happens to hemoglobin A1c, glucose levels, and body weight, that you get not synergistic, an additive effect between the GLP-1 receptor agonist and the SGLT2 inhibitor on glucose levels and body weight. So my interest was in measuring ketones under these circumstances, because if you increase ketogenesis, but at the same time you give something that stimulates insulin, you should be canceling the effect of the SGLT2 inhibitor completely. Now, against this was the observation that in MPREG, as well as CANVAS, SGLT2 inhibitors were given on top. But unfortunately, they did not save the samples. And so I could not measure ketones in MPREG, probably going to do it in CANVAS, to test whether this was a cancellation of the effect on ketonemia. Now, if the liver does, now, we must consider the heart, right? So if the liver can make but not use beta-hydroxybutyrate, this will be exported. And where does it go? Everywhere. It goes to the kidney, it goes to the heart, and goes to the brain. And in fact, supply of ketones to the brain under conditions of prolonged fasting was proposed in the early 60s to be an evolutionary conservation mechanism. Now, apparently there was a time where our ancestors were hunter-gatherers. They would eat once a week or once every 2 weeks when they could shoot down a big animal to feed on, and in between there was prolonged fasting. And Cahill was the first in the 60s, exactly in 65, to propose that the rise in keto bodies, which they actually measured by fasting volunteers for 28 days, today it would be completely unacceptable. And they measured ketone levels that obviously rose steadily well into the millimolar range, and they argued that this increase was essential for the brain to save on glucose utilization and to stay alive. But we will not talk about the brain, we'll talk about the heart. Now, what's the condition of the diabetic heart in general, metabolically? I'm going to concentrate only on metabolism. In the discussion, we can also talk about other things. So here we go. Under starter circumstances, so before treatment, the cardiomyocyte is exposed to an excess of fatty acids. Now, in the heart, there is plenty of GLUT4, which is the insulin regulatable transporter activated by insulin. So the big deal about metabolic flexibility or substrate competition is that in insulin-sensitive tissues, it is true that if one substrate is lacking, you can use an excess of the other to compensate and maintain energy production, with one major limitation, that the uptake of free fatty acids occurs down a concentration gradient, whereas for the transport of glucose, you need insulin regulatable transporters. And if there is insulin resistance, this process is down in patients, with, for example, with type 2 diabetes. So not only is there an excess of fatty acids being presented to the cardiomyocyte, but there is also a difficulty for glucose to be transported into the myocardiocite to produce the pyruvate, which is then used in the TCA cycle for energy production. This is the condition of the diabetic heart at baseline, before you do anything. Not only that, but there is a lot of basic physiology and energetics that will explain why the oxidation of fatty acids is energetically less efficient than the oxidation of carbohydrate. This is because of an element of the inner mitochondrial membrane depolarization. It's a problem with superoxide production in the energy transport chain, and even the activation of transport of calcium. So without going into details, it's long been known, and there is an extended literature, that if you look at the molecule of a free fatty acid, it has less oxygen than a molecule of carbohydrate. So the oxidation for the production of the same amount of ATP requires more oxygen for lipids than it does for carbohydrate. And there is also the problem that because of these mechanisms, the oxidation of free fatty acids is inefficient. The ATP that is produced is partly dissipated as heat, and there is the upregulation of proteins such as the uncoupling protein in the mitochondria. This is, again, basic physiology. And is there insulin resistance in the heart? Yes, there is. If you map the insulin receptors at the whole body level, you find an accumulation in 2 or 3 places. One is the liver, and the other one is the heart. So the heart, the cardiomyocyte, is an exquisitely insulin-sensitive tissue. These are studies that we carried out almost 20 years ago, actually more than 20 years ago, where we used euglycemic insulin clamps, and we measured skeletal muscle glucose uptake over a range of insulin sensitivities, but also myocardial glucose uptake using PET scanning in conjunction with the insulin clamp. And as you can see, when there was insulin resistance at the whole body level, this went with insulin resistance in skeletal muscle and myocardial glucose uptake. So no question about that. Not only that, but we also found this weak association between myocardial glucose uptake and the ejection fraction. So the lower myocardial glucose uptake, the lower the ejection fraction. Now, you don't know which is head and which is tail, but this is just one association. And we also looked at myocardial glucose uptake in patients without diabetes but with coronary artery disease, in patients with diabetes but without coronary artery disease, and in patients with both diabetes and CAD. And we found a uniform reduction in myocardial glucose uptake. So there is insulin resistance in the heart. Consequently, there is inhibition both in terms of glucose transport, as I mentioned, because this is GLUT4, as well as in the generation of acetyl-CoA from pyruvate, because the rate-limiting enzyme, which is pyruvate dehydrogenase, is inhibited by products of free fatty acid oxidation. So the heart is under discouraged conditions because of the insulin resistance and because of the excess free fatty acids, I will rush on, to use rather free fatty acids than glucose under the circumstances. Now, in humans, and this is again studies that we did a long time ago with catheters across the heart and measuring coronary blood flow, beta-hydroxybutyrate in the fasting state is a preferred substrate by the heart. These are fractional extraction rates. So this is the ability of the heart to extract fractions of the delivery of each of these substrates, free fatty acids, pyruvate, lactate, and glucose, under fasting conditions and then during an insulin clamp, showing that in the fasting state, the heart is the organ in the body with the top avidity for beta-hydroxybutyrate. It likes it as a feed. And even if you do then atrial pacing, so if you impose something that looks like exercise, you see that when you increase the rate pressure product, oxygen consumption increases. Of course, this is myocardial oxygen consumption, but the fractional extraction of beta-hydroxybutyrate is constant. So the heart continues to use lots of beta-hydroxybutyrate when you exercise. Now, this is the comparison with the forearm, but that will take us away. Then there is biochemistry. And this is the energetics of glucose, palmitate, and beta-hydroxybutyrate. And if you just concentrate here on the oxygen cost of calories, how much oxygen is required to produce the same amount of calories, you see that beta-hydroxy competes with glucose, but both are better than palmitate. And if you look at the cost of calories per C2 for every 2 carbon products of oxidation, you see that even that beta-hydroxybutyrate is now even better than glucose and far better than palmitate in producing energy. So this is, again, biochemistry that applies everywhere. And the heart must take advantage of this. So here comes the rescue, beta-hydroxybutyrate, which is transported through these monocarboxylic acid transporters 1 and 2. It's a facilitated transport, and it does not require insulin. So glucose does, but beta-hydroxy doesn't. So if the levels increase, all tissues will be exposed to more, and all tissues that express this glucose transport, these monocarboxylic acid transporters, which are ubiquitous, they are found in many tissues, more beta-hydroxybutyrate will be taken into the myocardial site. There is immediate production of an NADH, which immediately goes into oxidative phosphorylation, and then there is preferential use of beta-hydroxybutyrate over both free fatty acids and glucose. This is where the hypothesis becomes hypothesis in humans. Why is that? Because there are a number of other roles, and again, there is a literature that is really challenging to review, but beta-hydroxybutyrate is believed to do loads of other things, and you can go through the list. But this is a study that was done recently and done by a group in Denmark, where they tested the hypothesis that beta-hydroxybutyrate can compete with glucose. So what they did, they took a group of healthy volunteers, they did a control study during a clamp with PET, and then they did a test study where they infused exogenous beta-hydroxybutyrate. And what did they find? They found, first of all, that compared to saline infusion, increasing ketone levels into the millimolar range, it must be said, but this was an acute study, led to an increase in blood flow in all coronaries, the left anterior descending, the circumflex, and the right. So this is the vasoactive effect of ketones that Richard was alluding to in humans. And concomitant with that, this is the image of a cross-section of the heart of these healthy volunteers at mid-heart level with FDG, which is an analog of glucose, under the influence of insulin. And you see there is very nice homogeneous uptake of FDG under control conditions, saline infusion, and there was a homogeneous reduction in glucose uptake when you raise the beta-hydroxybutyrate level. So this bit of the hypothesis, within the boundaries of the experiment that was done, which was acute and in healthy volunteers, has actually been verified. If you increase the provision of beta-hydroxybutyrate, you impede the utilization of glucose. So this is another kind of Randall cycle that has been brought to the forefront in humans. Finally, there is also this issue of how does the heart meet up an increase in energy, for example, during exercise. So the oxygen demand goes up and the oxygen supply must follow. And for oxygen supply, the main mechanism is the increase in coronary blood flow, and we call that coronary reserve, and the cardiologists use this as a test to explore microvascular function in patients with cardiac disease. But there is also an element of increase in oxygen delivery to the heart, which is carried by hemoglobin. So the basic equation in myocardial metabolism is that myocardial oxygen utilization is a product of blood flow and the ability to extract oxygen. So what, at least in the study that we looked at, what the beta-hydroxybutyrate infusion, exogenous, acute, into the millimolar range, what have you, was able to do was to increase coronary blood flow. But we've known from the very beginning that SGLT2 inhibitors consistently increase the hematocrit. So if you take 1 ml of blood after treatment compared to before treatment, you have relatively more hemoglobin after treatment than before. Hemoglobin carries oxygen, and therefore the delivery of oxygen to tissues is improved. This is the data, for example, from EMPEREG. You see that already at the first visit, 12 weeks after initiation of treatment with either dose of empagliflozin, there is this rise in hematocrit which is persistent. It's incredibly consistent. It's almost a measure of compliance, of drug compliance. And if you stop the drug, it comes all the way back down. I will skip this. And there is also this observation that has been confirmed in at least a couple of other studies, that following treatment with an SGLT2 inhibitor, there is a slight and probably transient, but statistically significant, increase in erythropoietin. We don't know what the mechanism is for this, but there is this fact. So here we go. The hypothesis now is that because of the status of type 2 diabetes, on top of which is SGLT2 inhibition, there is increased lipolysis and free fatty acid delivery to all tissues in the body. There is, because also of the reduction in the insulin to glucagon ratio, stimulation of ketogenesis, export of ketone bodies, at least from the liver, possibly also from the kidney, increased uptake of beta-hydroxybutyrate by a non-gated mechanism that doesn't require insulin and does interfere with insulin action on glucose. There is increased utilization preferential to glucose within the myocardial at the same time as there is a better delivery of oxygen to the heart. Now, the part of the hypothesis that has not been demonstrated in humans is this bit here. Are we also concomitantly able to block the oxidation or even the transport of fatty acids into myocardial if we provide an excess of beta-hydroxybutyrate? That's part of the hypothesis, but it's not been verified. So this bit has been verified last year. Here, we are doing a study to see whether this could possibly be the case. So theoretically, you are doing a metabolic service to the heart, who's already in difficult metabolic conditions, by delivering more oxygen, providing a salvage or rescue substrate, and improving the energetics of ATP production in the mitochondria. The big question, and here I think comes the main hypothesis, is how strong is this mechanism? Is this sufficient to produce the cardiovascular protection that has been seen in EMPEREG and CANVAS, and according to the press release, also in the DAPA, in DECLARE? This we do not know. We cannot partition in vivo how much was the contribution of the diuretic effect, how much was the contribution of the reduction in blood pressure, on the background of less hemoglobin A1c and reduced body weight, and how much could possibly be the contribution of this metabolic shift. Now, I will conclude by saying, is there a way of partitioning the effect to the different mechanisms. And I started out by saying this or any other hypothesis is not exclusive of the others. There is probably a convergence of all these mechanisms: reduction in preload, reduction in postload, amelioration of the energetics and substrate utilization. How much of which is responsible for how much of the... I don't know that this is humanly possible to demonstrate. So eventually the hypothesis is in the big picture undemonstrable. We can only say that bits and parts of it have been documented. Some are going to be further documented, but the big thing is probably very difficult, with one exception. And the exception that I would like to submit to you is that unexpectedly, completely unexpectedly, the major finding in MPREG and Canvas was the reduction in hospitalization for heart failure. Now, the failing heart, and the next speaker is going to expand on that, the failing heart is under very difficult metabolic and energetic conditions. But it is striking that whereas we have no treatment really other than beta blockers, diuretics, and ACE inhibitors for heart failure, which is exploding in type 2 diabetes, both in the version with reduced ejection fraction and in preserved ejection fraction form. It is interesting that in both the trials, and we will see what Declare will show, there was this striking protection against hospitalization, and by inference, we must say also against the mortality from heart failure, or at least the morbidity in patients with heart failure, whether they have it at baseline or not. And this is the reason why they are now starting trials which, rather than looking at MACE, which is a composite endpoint, are specifically looking at heart failure. And I would submit to you that the only cardiovascular disease that is specific to diabetes, above and beyond the atherosclerotic involvement of the coronaries, is actually heart failure. And so the energetics, the substrate utilization, and the potential impact of these mechanisms in heart failure is now what's on the table to be tested by many investigators, including many colleagues in cardiology, for the next few years. And with that, I apologize if I went longer, and thank you for your attention.